Hello everyone, welcome back to our channel. I'm Dr. Ashmita. In this video, we'll continue with the G-protein coupled receptors. In the first part of this video, we have discussed about the G-protein coupled receptors, the basic introduction. What are they? What are G-protein coupled receptors? What do they do in the GPCR signaling? That is the cell signaling and the basic structure of the GPCR signaling pathway that includes the receptor, that is the serpentine receptor which traverses the cell membrane 7 times, hence also called the 7 transmembrane receptor. Upon this ligand binding, how it transmit the signals to this coupled, the G protein coupled receptors. We are calling it G protein coupled because it is in coupling with this G protein complex, which is composed of alpha, beta, gamma subunits. Whenever signal is transmitted to the G protein, it will in turn activate the effectors. Now, how this effectors will cause the generation of secondary messengers final and leading to final output. So this is the basic structure of the GPCR signaling pathway. Let's discuss in detail about the mechanism of GPCR signaling. So consider this first image showing you the cell membrane over which there is a serpentine receptor traversing the cell membrane 7 times, and it is bound to this G protein complex. The G protein complex formed by the G alpha subunit, beta subunit, and gamma subunit. Alpha, beta, gamma forming this heterotrimer. Three different is this hetero, and trimer means three subunits. And the alpha subunit, it is like attached to this inner surface of cell membrane with the help of this lipid tail. And this alpha subunit, it has two kinds of domains. Firstly, GTPase domain, second helicase domain. These are the two domains of the G alpha subunit. The GTPase domain, it has a groove in it. This is the groove. This is the groove of the GTPase domain where the guanine nucleotide, the GDP or the GTP will be bound to this alpha subunit here. So this GTP in inactive state, whenever there's no receptor signaling, whenever there's no ligand, no extracellular signal, it is in inactive state. This receptor is inactive, even this is not stimulated. In that inactive state, the GDP, guanosine diphosphate, GDP will be attached to this groove of the GTPase domain, and this GDP is prevented from falling off by this helical domain. So the helical domain will act like a lid. It will cover the GTPase domain. So it is like a lid covering the GTPase domain and prevent this GDP from falling off in inactive state. So this is about the receptor, the alpha, beta, gamma subunits in inactive state. Whenever there is a receptor signaling, that means there is an extracellular signal coming in the form of a ligand. This will lead to the receptor signaling, which is transmitted via this intracellular loop to this alpha subunit. So here this ligand will bind to the extracellular loop. Ligand is binding to the extracellular loop here. It will lead to the signal transmission all via these loops, and finally the intracellular loop, which is connected to the alpha subunit, it will receive this signal. Because of this signal, there is a conformational change. There is certain conformational change of the alpha subunit. Conformational change means three-dimensional structure change. That is change in the 3D structure of this alpha subunit. Because of that, this helical domain will be lifted off. So it is usually in inactive state, it is covering like a lid. Now this lid, it is lifted off. When the lid is lifted off, now this GDP, inactive state, the GDP was bound here. It will be exposed. It will fall off. That means GDP is out and GTP, abundant of GTP, guanosine triphosphate. It is abundantly present in the cytoplasm. GTP abundantly present in the cytoplasm will diffuse into the alpha subunit and sit inside this groove, sit in the groove of the GTPase domain. Whenever this GTP is bound here, now this alpha subunit is called an active. It is an active G alpha subunit. The GTP bound alpha subunit is said to be an active form. Because of this activation of the G alpha, it will separate off from this beta and gamma. So the beta and the gamma subunits will stay together like a dimer. They will stay together like a dimer bound to this plasma membrane yet. Gamma is tightly bound to the plasma membrane and beta will stick to the gamma subunit. Both they will stay together as a dimer. Only the alpha subunit is separated out. And this separated G alpha subunit now it will lead to the effectors, the activation of effectors. So the kind of pathway it will choose, it will depend upon the type of alpha subunit. The alpha subunit can be S. S is a stimulatory. S is a stimulatory subunit or G alpha I. I means inhibitory. It is an inhibitory subunit or G alpha Q or G alpha 12 by 13. 12 or 13 subunits. So the whatever the pathway it has to choose, it will depend upon the type of G alpha subunit. The type of G alpha subunit that is activated, if at all it has like chosen the stimulatory pathway, it depends upon the type of ligand here. It depends on the type of ligand or the type of receptor being activated. The ligand and the receptor activation will decide the pathway of the G alpha subunit, whichever it has to choose, stimulatory or inhibitory or the Q or 12 or 13 pathway, it will be decided by this ligand and the receptor. And one more thing, here this we are calling it as a GTPase domain. We are saying it as a GTPase domain. The normal function of GTPase is to the GTPase is to hydrolyze the GTP. That means it will convert GTP into GDP. Hydrolyze. We are calling it a hydrolysis. It will hydrolyze, it will remove phosphate from the GTP and bring it back to the GDP state. So here we are like GTP is coming and binding in active state, the GTP is coming and sitting in this groove. And as soon as the GTP sits, we are calling it as an active. When GTP is bound, it is active. Active G alpha will separate it off. Now this it is inherently it has this inherent GTPase activity, but it is not able to like hydrolyze it. That means this separation of like it will take milliseconds to seconds of time. So within this milliseconds to seconds, this GTPase is unable to hydrolyze the GTP because it takes much longer. GTPase will take much longer to hydrolyze the GTP. Therefore, as soon as this GTP is coming and sitting, it will instantly become the active G alpha subunit. As soon as GTP is bound to this G alpha, it will instantly separate it will instantly separate off from the beta gamma dimer. And this GTPase, it will take time to act to hydrolyze the GTP. Within this time only, within this milliseconds, fraction of milliseconds to seconds, the G alpha active G alpha subunit, it will choose this pathways to act on it. And coming to this, which pathway it is decided by the ligands and the receptors. So let's see a few examples of the ligands and receptors which stimulate the G alpha S pathway. So examples are divided into hormones. The ligands can be hormones, neurotransmitters, local mediators, or metabolic ligands. Among the hormones, the TSH, thyroid stimulating hormone, is a ligand which acts on this TSH receptor, number one. Or it can be a parathormone acting on PTH receptor. Number three, luteinizing hormone acting on luteinizing hormone receptor. Fourth is FSH, follicle stimulating hormone acting on FSH receptor. These are the examples of few ligands and the receptors that stimulate the G alpha S. Let's see the neurotransmitters like dopamine. Dopamine is a ligand acting on D1, D5 receptors. Local mediators like histamine. Histamine acts on the H2 receptors. Prostacyclin. Prostacyclin is the PGI2, which act on this IP2 receptors. These are like the examples of ligands and receptors that stimulate the G alpha S, and metabolic ligands like GHRH, that is a growth hormone releasing hormone acting on the growth hormone releasing hormone receptor. They stimulate the G alpha stimulatory pathway. These are the examples of ligands and receptors. So now let's begin with this pathway of G alpha S. G alpha S stimulatory pathway, let's say it. So we have studied there is this GPCR, this receptor. Upon this ligand binding, the signal is transmitted. The signal is transmitted to this G alpha subunit. G alpha subunit, GDP exchanged for GTP. Now it is called an active GTP. It is called an active G alpha subunit. Now this active G alpha subunit has chosen this path, G alpha stimulatory pathway, because of these kind of receptors and ligands. Now on choosing this pathway, whenever this G alpha subunit, the stimulatory type has been activated, it will activate the effector. Next step is effectors. After this is the effector. The effector here is adenylyl cyclase, AC. Adenylyl cyclase is the effector. It gets activated. When this adenylyl cyclase gets activated, it will convert ATP to cyclic AMP. This adenylyl cyclase will convert the ATP to cyclic AMP. So there is lots of generation of cyclic AMP, which are the secondary messengers here. So we have discussed the basic structure. First, receptor ligand binding, receptor activation. Next, the G protein gets activated, that is the G alpha getting activated. From the G alpha, there's effector activation. In case of stimulatory, this adenylyl cyclase is the effector. The effector will lead to generation of secondary messengers. Here it is the cyclic AMP. Now this cyclic AMP, now it will finally act on certain kinase enzymes. The kinase enzymes here are this pKa. pKa is a kinase enzyme. It will activate the pKa enzyme. pKa enzyme, this kinase, will contain, there are two regulatory units and two catalytic units. So here R means regulatory. This is the regulatory unit. C is for catalytic unit. R is for regulatory, C is for catalytic. In inactive state, whenever the pKa is inactive, it will contain, there are two R's, two C's which are bound to each other. Regulatory is bound, the regulatory is bound, this regulatory, it is bound to the catalytic. It is in bound state, inactive. Whenever this cyclic AMP, cyclic AMPs are produced, the cyclic AMPs will enter the pKa and it will bind to the regulatory subunit. So for every one regulatory unit, there are two cyclic AMP binding sites. That means whenever there is for two regulatory units, four cyclic AMPs will be bound. So there are two cyclic AMPs on each regulatory. Because of this binding of the cyclic AMP to the regulatory subunit, the catalytic unit, this is the catalytic unit, it is now free. It is in unbound form. It is free now. It is free. Free means it is active kinase. Now it is in active form and it has this kinase activity. Kinase means kinases are those which add phosphate group. That will add the phosphate group. It will add phosphate from where? From where it will take the phosphate? It will take the phosphate from the ATP and add it to target proteins at what? At the serine or the threonine residues. They will add the phosphate at the serine or threonine residues. So we are calling it proteins. These are proteins. Proteins are made up of amino acids, one of which amino acids are the serine amino acid or threonine amino acid. So this kinase is the catalytic unit will act like a kinase. That means they add phosphate to the serine or the threonine residues of the target proteins here. The examples of target proteins here are the L-type calcium channel present in the cardiac muscle is one example, or this glycogen phosphorylase kinase enzyme present in liver and muscle. This is the other example. Or you can take the CREB. CREB is the transcription factors present in the nucleus. These are the few examples of the target proteins that are activated by these catalytic subunits. And this catalytic subunits, it will choose the specific targets. That means it will choose which this one, or this one. There is specificity. This specificity is brought about by this AKAPs. AKAP means A kinase anchoring proteins. A kinase anchoring proteins present just under the cell membrane or the mitochondrial membrane or in the nucleus. Because of this AKAPs, the catalytic subunit is brought like chemotaxis, how they are attracting there. Here also, because of AKAPs, this catalytic subunit is targeted, it is pulling towards the specific locations. So it will prevent a random phosphorylation, not that every molecule or every protein will come in contact, it will not phosphorylate. Randomly, it won't go on phosphorylating everything. It will target specifically. That specific targeting is brought about by AKAPs, that is the A kinase anchoring proteins. So this is called the localization and compartmentalization. This pKa activity is localized by the AKAPs, that is A kinase anchoring proteins. It will tether. Tether means it will like hold the pKa, that like a bond or a glue. It will like stay with the help of this thread. It will tether the pKa to specific subcellular sites like membrane, mitochondria, nucleus. It will prevent random phosphorylation. So this is about the AKAPs, that is, it will cause localization. After targeting these specific proteins, for example, L-type calcium channel, let's see this example in the cardiac muscle, it will increase the permeability of calcium. That means there's increased calcium influx. Calcium influx will increase. It will cause strong contraction. In the cardiac myocytes, calcium influx is increased, will cause strong contraction. Other example is glycogen phosphorylase kinase enzyme present in liver and muscle, which will increase this glycogen breakdown. Whenever glycogen breakdown is increased, there's glucose release into the blood, increased glucose release. Third example, let's see this transcription factor present in the nucleus, that is CREB. This CREB, this active C catalytic subunit will enter the nucleus. This catalytic subunit here, it will enter this nucleus. Catalytic subunit of this pK enzyme, it will enter the nucleus and it will phosphorylate this CREB. CREB is present inside the nucleus. CREB is a transcription factor present in the nucleus. It will phosphorylate the CREB on serine residue 133 is the sequence at serine 133. This phosphorylated CREB, this is the phosphorylated CREB. Now it will bind to DNA at CRE. It will bind to this DNA at the CRE. That is the CREB response element. This is the response element. This is the response element. And it will also recruit other co-activators. Upon binding to this response element, it will add on other co-activators and cause transcription. It will initiate transcription. Transcription means the DNA becoming the mRNA. mRNA finally translated into protein, the final outcome. That is about this nuclear transcription factor, CREB. So, because of this transcription of genes, it will release genes for growth and metabolism. Final output is growth and metabolism. So, let's revise again the mechanism. Because of ligand binding, signal is transmitted to the alpha subunit. The helical domain lifted off, GDP out, GTP present abundantly in the cytoplasm will move in, and this is called active GTP, active G alpha. If it is bound to GTP, it is called active G alpha. If it is bound to GDP, it is inactive. It is inactive G alpha. Because of this GTP binding, it is called an active G alpha. Active G alpha will have many pathways, stimulatory, inhibitory, Q or 12, 13 pathways. And whichever pathway it has to choose depends on ligands and receptors. Once it has decided to choose this alpha stimulatory pathway, the stimulatory, it will cause increase, it will cause activate the adenylyl cyclase, which is the effector here. The adenylyl cyclase will lead to generation of secondary messenger cyclic AMP here. Cyclic AMP will now activate the phosphokinase A. That means the cyclic AMP will bind to the regulatory subunit. Catalytic subunit, which is the kinase domain, is free to act. The kinase will add phosphate to the target proteins and the serine or threonine residues. The target proteins here are examples L-type calcium channel of the cardiac muscle, glycogen phosphorylase kinase enzyme of the liver muscle. Thirdly, there is transcription factors, CREB, transcription factors of the nucleus, and the specific localization, the specific localization of the membrane, mitochondria, or nucleus brought about by this A kinase anchoring protein. This is about the GPCR signaling. After the final outcome, this transcription of genes or this contraction or the glucose release, whatever the actions has to be delivered, the final actions after being delivered, now the signal has to be terminated to prevent overactivation. It is terminated by the cyclic AMP degraded by this phosphodiesterase. Here, at this step of the cyclic AMP cyclic AMP produced by this adenylyl cyclase, the cyclic AMP will be degraded because of phosphodiesterases. Diesterases means it will break down cyclic AMP. When there's no cyclic AMP, this pathway doesn't occur. So, the cyclic AMP degraded by the phosphodiesterases, the AMP or the R subunits rebind. Now this R subunits will rebind to catalytic subunits, inactive RCTC. When there are no cyclic AMP, this catalytic subunit will bind back. It will be in bound form to the regulatory subunit. That means in inactive state. Inactive state means there's no targeting of these proteins, no signaling happening. Other thing is by this protein phosphatases. Phosphatase means it will remove the phosphate. It will remove the phosphate from target proteins. Target proteins here. So here is the phosphatase acting on this target proteins where it will bind to serine and threonine. The target, it will remove the phosphate groups from here. Because there's no activation here, this pathway, the final output is not happening. The final output does not happen. So this is about how the signal termination occurs. The signal termination is essential to prevent this overactivation. Signal does not have to act all the time. So once the final output is delivered, it has to end. To prevent overactivation and restore the baseline signal, optimal signaling should be present, not overstimulation. For that reason, signal termination is a must. And let's see a few clinical examples with significance. So firstly, we'll take cholera toxin. The cholera toxin will it will inhibit the GTP hydrolysis. That means here GTPase domain is present, which will cause the hydrolysis of GTP. Once the GTP hydrolysis occurs, now the GDP will come back. That means it will return to this inactive state. If this hydrolysis is not occurring, that means always continuously there is a GTP present in this groove of the GTPase domain. That means continuous activation of G alpha, continuous signaling will occur. So cholera toxin will not let this happen. Cholera toxin will not let the GTP hydrolysis to occur. It will cause continuous stimulation. Continuously GTP will present. Continuously G alpha will be stimulated. So the G alpha, that is the cholera toxin, will inhibit the GTP hydrolysis and it will cause constitutively active adenylyl cyclase. Increase in cyclic AMP. Here mechanism we have studied. So once this is continuously active, continuously AC, the effector will be active, continuously secondary messengers are generated. So because of which final output is increasing chloride and water secretion, which will lead to secretory diarrhea. So this is the clinical significance of cholera toxin that will continuously cause G alpha stimulation because it will inhibit the GTP hydrolysis. Second example is TSH, the thyroid stimulating hormone receptor mutation. Because of receptor mutation, even in the absence of a ligand, the receptor is continuously active, continuous signaling is occurred. So this constitutes activation of G alpha stimulatory pathway. That means continuously cyclic AMP signaling occurs, because of which there is continuous increased thyroid hormone that will lead to toxic thyroid adenoma. Let's see the third example, luteinizing hormone receptor mutation. That means because of receptor mutation, even in the absence of this luteinizing hormone ligand, the receptor is continuously active. So continuous G alpha S activation There's overstimulation of steroidogenesis. All these hormones are the steroids which are continuously generated that will lead to testicular or Leydig cell hyperplasia. Coming to the GNAS gene mutation, because of this GNAS gene, it will normally encode G-alpha stimulatory subunit. Whenever this gene is mutation, the G-alpha-S is continuously active. That means uncontrolled growth hormone release, because of which there is acromegaly. Overstimulation continuous growth hormone will lead to acromegaly. Fifth example is beta-adrenergic overstimulation. Beta-adrenergic receptors are overly stimulated, because of which there is increased G-alpha-S stimulation, because of which there is chronic cyclic AMP or the PKA signaling pathways continuously active. That means here, this is the example of cardiac muscle. Because of this permeability, because of activation of this channel, this continuous calcium influx, it will cause strong contraction. Whenever the signaling is continuously active, the stronger, more and more continuously stronger contractions happen, because of which this maladaptive remodeling. It will lead to cardiac arrhythmias and hypertrophy, because the ventricle muscle will increase in size because of strong continuous contractions and cardiac arrhythmias. These are a few examples of the continuous GPCR signaling. When the signal termination doesn't happen, or because of any mutations of the toxins, the GPCR continuous signaling will lead to these pathologies. So this is about the mechanism of action of the GPCR signaling, especially this is in reference to the G-alpha stimulatory pathway. In our next video, we'll continue with this inhibitory and the other pathways. So if you find this useful, please like, share and subscribe to our channel. Thank you.